There's a revolution brewing in the field of functional genomics. A new CRISPR-based technology called HACE allows scientists to do continuous mutagenesis in endogenous loci.
Functional genomics aims to understand what each DNA sequence does. One of the tools that are most often used for that is mutagenesis, which consists of mutating different bases in a sequence and characterizing the effects of each mutation.
This can allow us to identify genetic variants that, for example, tune gene expression up or down or produce proteins with different characteristics. But how do we mutate single bases?
So far, mutagenesis screens using base editing have been limited by two factors. First, each different mutation to be tested required a different gRNA, which meant producing and transfecting all of those gRNAs.
In addition, each gRNA only helps cover a small region of at most 50 base pairs. That meant that some genomic regions could not be covered simply because there were no good gRNAs to target them. And of course, introducing more than one mutation at the same time was complex.
So the dream was being able to introduce one or multiple mutations in a large part of the endogenous locus. And thanks to the group of Fei Chen at the Broad Institute, the dream just became a reality.
For this, the team combined 3 different proteins to build a new gene editing system called HACE. First, Cas-nickase is used to drive the complex to the desired target site, of course, using a gRNA. Second, a deaminase enzyme that introduces the mutations, just as with base editing. The trick, however, is on the third component, a helicase protein.
Helicases are proteins that wind and unwind the DNA in our cells. By attaching a helicase to the complex, the scientists managed to get the gene editor to attach to the DNA at the target site and quickly unwind it as the helicases moves forward.
And while the helicase is taking its stroll, the deaminase is hard at work, mutating the bases as it passes them. This leads to the efficient introduction of mutations in a range of up to 500 base pairs from the target site. That's a 10 times larger editing range.
The raw rate of mutagenesis of the system ensures that for each kilobase, 1 to 5 mutations are inserted. This allows the characterization of single or multiple mutations along a genetic sequence.
The best part about this system is that it is completely tunable. By changing either of the components, it's possible to modify the range, frequency, or even the type of mutation introduced.
And the authors showed the amazing flexibility of HACE by applying it to different problems. For example, they introduced a mutation to the MEK1 gene in cancer cells to see which mutations made them resistant to MEK inhibitors.
Another application they showed was finding mutations in a splicing factor that can cause missplicing. And they also used this mutagenesis approach on regulatory regions to see which bases are important and how mutations affect gene expression.
The HACE system promises to revolutionize the way we do functional genomics. Not only it will facilitate mutagenesis, but it can also be applied simultaneously to various genes.
And we know you'll be concerned about off-target effects, and of course the authors were too. Good news, HACE has only very mild off-targets, and they're all because the deaminase enzyme is a bit too generous with its love, but that's fixable.
We can wait to see what other application HACE will be used for in the future, but we are sure this is a name we'll be hearing a lot.
We here at With CRISPR will follow this topic closely and be ready to tell you all about it.